So next up then, we will look at the liver. And the liver is a very large, reddish brown, I'd probably call it more brown organ, located in the upper right, more of the right. It covers quite a bit of the top of the abdominal cavity, and it is the largest internal organ. Typically, students who have not dissected anything before are surprised by how large the liver of any animal actually is. So your liver is a large organ. It is divided. It has a right lobe and a left lobe, and then it's enclosed by a capsule that surrounds it. Each lobe is then separated into hepatic lobules. So whenever you see HEPA, HEPA, that always deals with liver. These hepatic lobules consist of hepatic cells, and there again they're going to form a vein or a tube that's going to drain into the gallbladder. It's going to drain down by the pancreas. Hepatic sinusoids are separate groups of hepatic cells. So, you know, in biology we talk about cells build tissues, tissues build organs. These hepatic sinusoids are groups of hepatic cells that are going to work together. Blood that comes from the hepatic portal vein carries blood rich in nutrients into your liver. You know, they say that if you eat liver, which I've tried liver, if it's like burnt, I'll eat a little bit of it. It's not something that I'm going to want to eat all the time, but it is very rich in nutrients. Liver is very good for you. Because it's full, this brings us the blood that's rich in nutrients is going to go into your liver. It's going to go from the sinusoids, those little groups of cells, into the central vein, and then it's going to exit the liver by the hepatic vein. So hepatic portal vein brings basically blood-rich nutrients to the liver, and then it's going to exit through the hepatic vein. Kupffer cells carry on phagocytosis in the liver. Your liver is very important for getting rid of dead, bad cells, things like that. Alcohol, cirrhosis of the liver. Your liver helps to break things down, so that's what phagocytosis is. And if you see a liver that has cirrhosis, a lot of times when we go to the cadaver, you'll see a regular liver and one with cirrhosis, and it just has like almost corroded parts that are missing from it. Secretions from the hepatic cells are collected in bile canals that converge to become hepatic ducts and finally the common hepatic duct. So this part up here deals with blood. This is how blood flows through the liver. Down here, we're actually talking about secretions that are formed from the liver. So bile is one of the main things. It goes through canals, forms the hepatic duct, and then the common hepatic duct is going to be the large final one. So here you can see the lobes. The right lobe is larger than the left lobe. Gallbladder is going to store bile. So the liver produces it. It is stored. The excess is stored here. And then I can see here's a hepatic duct, here's a hepatic duct, and then it's going to come down into this common bile duct that is right there. And here I can see the vein and arteries that are coming also into the liver. Do notice all of them enter at pretty much the same area. Here is just a microscopic view, so you're looking at the inside. Probably won't have this on the test, but you can see the small central veins that run through. This is the sinusoid, so that's that group of cells, so that you can see what it looks like. And then you're just going to have all sorts of these clusters that fill the liver. So your liver, if you cut into your liver, which we noticed when we dissected the cats, it's a solid organ. It's not like your stomach where there's a cavity or opening in the middle. The liver is pretty much solid cells that you're going to cut through. There again shows, you can see, so here's a Kupffer cell. That's going to get rid of its phagocytosis, going to destroy things. Here's the hepatic portal vein, the hepatic artery. There again, not a diagram that will probably be on the test. Functions. Your liver is very, very important, and if you have blood tests, very often they're going to test for liver results from your blood tests also. Metabolic activities, so breaking down, metabolizing carbohydrates, lipids, and proteins. It stores many things, glycogen, vitamins A, D, B12, iron, which is why you can overdose on some of these. Iron, you don't want to take in too much iron because it's going to be stored. It's not going to be released. Synthesizes, it's going to make, so here it's breaking down carbohydrates, lipids, proteins. Here it's storing, here it's making. So synthesize means to make. It's going to put together lipoproteins, which is a conglomerate of a fatty type of protein, phospholipids, and cholesterol. So your liver is what helps to produce cholesterol. Also, it converts carbohydrates and proteins to fat, which is necessary for energy and also storage if we're not using up all of those carbohydrates and proteins. Deaminates, which means it gets rid of part of amino acids. It's going to take the amine branch. This would be a chemistry type thing. It's going to take the amine branch off of the amino acid, and that forms urea. And urea is then also found like in urine. It kind of has that odor. It also synthesizes plasma protein and clotting factors. So people with hemophilia, sometimes their issue, the cause of their problem actually comes from something in their liver, because that's where some of the clotting factors come from. So your liver, besides just breaking down carbohydrates, lipids, proteins, it has a lot of other functions as well. One of the ones that you might have known already would probably be these two. It filters the blood. It cleans, kind of cleans the blood. It gets rid of damaged blood cells, foreign substances, and toxins. So toxins, here's where it helps to break down alcohol. And that's why cirrhosis of the liver is associated with too much alcohol. Damaged red blood cells, it's going to get rid of them. There again, those Kupffer cells are going to engulf them and digest them. And then the main role as far as our digestive system, it's going to secrete bile, and that is the liquid that helps to break down proteins, fats, and carbohydrates. So here's just a table that kind of puts it all together. You can see all of the jobs that we already went over. And bile itself is a yellowish green. If you've dissected any type of animal, as long as that animal was alive, that gallbladder usually has a greenish color to it. Fetal pigs, a lot of times fetal pigs are dissected, and the gallbladder won't be green yet because they have not eaten anything. So bile, yellowish green liquid. Hepatic cells are going to produce this liquid, and inside bile, most of it's water. There are bile salts, bile pigments, bilirubin and biliverdin, cholesterol, and electrolytes. These pigments, if you know someone who has liver damage, sometimes their skin coloring will actually take on almost kind of a greenish color. I mean, not like a bright green, but there's a tinge to people that have severe liver problems. Bile pigments then are part of the breakdown of red blood cells, and only the bile salts are the part of the bile that help with digestion. Gallbladder itself, kind of pear-shaped. It's always inside of the liver. It's connected to the cystic duct, which joins the hepatic duct, and that's what makes the common bile duct. Common bile duct you should know because that's where it's going to empty also into the small intestine. Here again, that sphincter muscle is going to control the release of bile. And what happens is between meals, the gallbladder stores bile, and then it's going to reabsorb water to concentrate that bile. And then when food passes into the small intestine, so after meals, when you have food present, that bile then is released from the gallbladder into your small intestine. So typically bile it's not going to always enter the duodenum. It's going to happen after a meal. This cholecytokinin is what stimulates the gallbladder to contract. So basically it's a squeezing motion that pushes the bile out of the gallbladder. Here again, the hepatopancreatic sphincter, same one that we talked about with the pancreas, remains contracted. It basically remains shut unless a peristaltic wave contracts by it, and that's when it's going to open up and bile will enter the duodenum. So this sphincter muscle, round muscle, is typically closed or contracted unless food is passing into the small intestine, and that's when it will relax and open and let bile enter the first part of the small intestine. So now, kind of the same picture. We did have the pancreas right here before. Now we're specifically looking at the gallbladder. So here's the cystic duct. It's going to come from both places. And then here is that common bile duct, which is the large one. And it's going to empty right with this pancreatic duct. And there is the hepatopancreatic sphincter muscle that is closed most of the time. Bile salts, the main thing that they do is to break down fats, emulsify fats into smaller droplets, absorb fat, and that doesn't mean that's not fat storage. This is actually taking fat into your cells. Cholesterol and fat-soluble vitamins, and that's what I said before. Fat-soluble vitamins are the ones you can overdose. You can take too many because it stores and builds up in your body. So the bile salts are going to absorb those things and store them for a period of time. And this emulsification allows lipase, which is the enzyme that breaks down fats, to digest fat molecules and break them down even further. And that's the end of the liver.